This presentation will be about so-called ambiguity codes that they were standardized about 50 even more years ago for molecular descriptors in molecular biology. Those ambiguity codes or incomplete specification of nucleotide coding was done due to the necessity to specify chemical structures of nucleobases and amino acid to have a chemical nomenclature. It was made by the International Union for Pure and Applied Chemistry and together with the same Union for Biochemistry. And there were several of those nomenclatures, but basically the one that was done in mid-80s is the essential one which stays, and we are using it nowadays. Biological systems are complex, and the good approach to them is to compare it with a computer system, especially since we have a lot of data arising from those complex systems which need analysis. And of course, as well, due to these problems, there's a necessity to standardize procedures that we could evaluate a large number of sequence data, whether it's in nucleotide or amino acid form. Unicode can be used to store those, and is used to store those. And the analogy is well seen because between the computer system and DNA. We have data from computers, strings, language. In DNA we have nucleotide sequence, amino acid, and proteome. The standard genetic code table itself translates the biological information from genotype to phenotype, and it basically represents a coding algorithm which shows the relations between RNA and the protein. Concerning this, we have 2 points. It's the table itself, the array, is characterized by two-dimensional arrangement of amino acids. However, those amino acids specified by a three-dimensional partition of codon triplets. So we see, let's go to standard nomenclature for amino acids. We have, do you have a pointer, João, perhaps? Do you have a pointer, maybe? Pointer. In addition to individual nucleotides, we have several symbols for purine, pyrimidine, amino, keto, strong and weak, and we have 4 specifications when A is missing, U, C, and G. They usually follow not G, H follows G, so it's H, and Y is for not U, and so on. In this way, we could more precisely specify some unknown or incompletely specified letters. And it is very important to say that for the nomenclature we have, in addition to rings, the most distant group from the sugar for keto and amino are taken into account in the discrimination. This sugar, this is most distant keto and amino groups. You will see later why this is important. And we will also I will show you how this nomenclature can be utilized in different calculations with the square opposition and Klein-4 group representation. This leads to not only to a better specification of elements, but it can be also used for specific calculation to obtain even physicochemical parameters from nucleotide sequence. The IUPAC nomenclature was used in during the last 30 years, for several models of nucleotide relationships or their hierarchy, and we see some of them. This is not surprising since we basically have 24 possible combinations of all 4 bases. So different groups, different researchers investigated and modeled different types of hierarchies. When I started to do it in 90s, I selected a relatively simple IUPAC nomenclature when using ring and amino and keto groups. So the small ring was given 0 and the large ring was given 1, and its less reactive group, keto, was given 0 and amino group was given 1. But basically, as I said, many researchers did. In this way, you can partition a unit interval to first in 2 letters concerning the ring, and then each half interval into 2 other more with respect to keto or amino group. So you have purine and pyrimidine rings with their groups, and of course a lot of 24 combinations and weightings. Another problem with the genetic code table, no, it's not a problem, it's characteristic, is that is basically not two-dimensional with respect to codons. It's 3-dimensional. So we have 2-dimensional arrangement of amino acids, but with respect to codons, we have tables which make rows, columns, and sub-rows of the 3rd base. So we used this to actually find out and calculate scores of codons and compare it to amino acid physicochemical characteristics. It is well known from the early papers that columns give a certain, quite a good actually, information about polarity, so of amino acids or physicochemical characteristics, while the rows up to a certain point connected to metabolic pathways and solubility, but not so strongly correlated. And of course, when we investigated all possible scores, all 24 scores, for the first base, second base, and third base, we found out that with respect to hydrophobic, hydrophilic parameters and lipophilic parameters, we found out that the first and the third base are not relevant. There were no statistical differences with respect to physicochemical characteristics of amino acids and scores, all 24 possible scores of amino acid weightings. But, however, UCGA, or its complement ACGU, and some other characteristics, UCAG is somewhat less than correlated, and we will see why this dichotomy. And some of those are not correlated at all, or poorly correlated. Our data actually showed that the best fit was with UCGNA, and that starting from the small ring and non-reactive group to the small ring with reactive group, then a large ring with non-reactive group, and then large ring with reactive group, it shows the best fitting to physical chemistry. Of course, it's also important to say that there are 6 possible ways to. You can start with 1, 2, 3 from row to column, or you can start from column to row. It's a classic 2, 1, 3 combination. And while the 2-1 combination, column row, is strongly correlated to all physical chemical parameters, which is of course natural because only second base is relevant. And in the model which we used to put it on the unit interval, you have a flipping coin model. So when you start fair coin, when you start with second base, you have 0.5, then the third base, the next base is 0.25, half of it. It's a classical model to place this on the interval. It is obvious that the columns carry the information even for nucleotides, physical and physicochemical information of amino acids. Now, the table itself is UCAG. However, it's the hierarchy is UCGA. UCGA is a hierarchy with respect to IUPAC nomenclature, because you have rings and you have groups. Now, this nomenclature, UCGA, fits well with Watson-Crick and Hoogstijn models for both strong and weak combinations. And you see that how they flip around amino or keto groups. So this is probably the reason why as we can see later, one can calculate with such arrangement of bases and rings. Interestingly, we bumped to this phenomenon somewhat later, analyzing Kimura's work. It's a very similar model to Kimura's model of from evolutionary biology of evolutionary distance. Here, 2 or 3 type model, which we will discuss later. A long time ago, in 90s, when I was introducing this UCGA arrangement based on IUPAC, I also showed that it incorporates so-called mutation rings. There are Shemion mutation rings and some others before. And as we will see later, those groups GU, GAUC are actually permutations of Klein groups within Cayley tables. Now, I will come to the problem of, it is not a problem, it's a rearrangement actually, of UCAG of classic table and reality, which is UCGA. The problem is in gray code. That problem of a gray code was addressed by Swanson in 1984. However, Swanson selected, as you can see here, classic definition UC by enumerating ring and group 1, 1 from G and 0, 1 for A. So this would fit the classic standard genetic code table. However, she admitted that there are other gray codes which could give arrangement of physical chemical parameters of amino acids, and that's basically what we did. In 2001, I did it, first published it in 1998. Of a gray code G01A11. It is basically due to reason that gray code can be observed in the context of so-called reflected gray code, when the element goes to a place to minimize distances. So basically, the element A, which is 1, 1, goes to the place 0, 1 to minimize the distance, to be 1-bit distance between each element. But it has changed the place, but it hasn't changed its symbolic value or informational value. So if you want to arrange elements with 1 bit difference, we have U00, C0, 0, 1, then goes A, which is 1, 1, and then it goes, the last one is G, which is 1, 0. So now from this you see how that it is written. So in the context of ring group IUPAC nomenclature, classic table is actually concerning rings and groups. Yes, it is coded in gray code, but the values are different. They are flipped for U and A. We published that a long time ago. And basically, this problem was addressed in a completely different manner by Martin Gardner in his mathematical column in Scientific American when he was speaking about gray codes. It's all written in that little paper about gray codes, and how it changes address, and how you flip the values and put the element with the value 1, 1 to the element with the place 0, 1 and flip them actually. Their places, but not their symbolic information or characteristics. So we have here this. This is, nothing has changed because amino acid also change with the blocks. It's the only thing that GNA flipped, but in three dimensions flipped first, second, and third base. It has no consequence to the table itself. But the problem arises if we are with the patterns. Some of the patterns, as we will see later, are not so obvious in the coded table. On the other hand, in some other engineering parts, gray codes of this type, as in classic table, are known as a standard map for Boolean functions. So Kamener maps is in gray code as a classic standard table, and for example, when it was standardized by the American Association in the 50s, at the same Congress, it was proposal of Wage to do it in a normal code. So according to Wage, it would be you can analyze Boolean functions as a decoded system, but the standard way of simplifying the calculations nowadays is so in this, I forgot to tell you that, of course, when you put it in a gray code, you can have a ring as well, as a Swanson got. So it is a classic ring, slightly modified. In a few moments I will show you how it actually looks. You see a reflected gray code FFLLS, so you have that FFLLS, so it goes like this actually. Till the end. And we can calculate by addresses, and we can of course go to process of coding and try to compress the information to, if we use one of the codes with the sliding blocks, for example linear block code of 6 letters, we could have blocks of 3 and 3 and analyze those blocks in the extended windows. Using, of course, the same methodology. This was all done in 2001, published. The adding procedure for classic linear block code, triple check code. Triple check is a good code because it has 6 digits as the code on 2 for each. Use the classic XOR algorithm and discriminates actually between strong and weak bonding. Yeah, in 2000 I first published this type of code and this model, and this is from 2001. when Pashko published this one, a new algorithm for the prediction of protein folding types, a classic code with 8 elements, so we can basically 64 are compressed into 8, so you can analyze dipeptides and code their symbolic bonds with only 64 elements instead of 3,700 something, which is inconvenient and not accurate when you have even larger data sets. Now, how it works, you see A columns and there, and it goes like this for a table. Either one. But of course, this is all in gray code, so the one I'm showing you is a classic standard gray code 1 for linear block triple check code. This gave a good error correction, up to 1 error out of 6. It's not perfect. Later on we showed that it works also for Hamming 4.7 code, so it covers 4 and the last, so it goes to the first ring of the second amino acid. It doesn't concern, but it is not, the Hamming code does not provide much help in this way, because all information necessary is contained within 6-digit information of each amino acid. At that time, we found out a very interesting problem, or characteristic of a code. In alpha and beta folds, because we used the Barton database and extracted sequences, at that time there was no sequence data, and there were a rather limited amount of reliable databases with well-defined structure by NMR or crystallography. So Barton did a good database, and we selected 140 alpha and 40 betas, which were reliable alphas and betas, and they were non-homologous. So, well, I spent some time extracting sequences. At that time, it wasn't so fast as today. It went very slowly. Anyway, when we analyzed, we did simple descriptive statistics, and we found out that it's not degenerate. Some of the alphas, when we have those codons with 4, 2, 6 amino acids with several codons, some were dispersed insignificantly different between alphas and betas, but some of the codons coding for the same amino acids were in alphas, some were in betas, and there was not a uniform grouping. Moreover, this depended on a dipeptide, as we showed 2 or 3 years later, and 3 peptides, whether they are in alphas and betas. So at that time, we realized that we are not speaking about a degenerate system. Some of the codons appear in some folds and the others in other folds, but despite of the fact that they are coding for the same amino acid. Now the problem of dipeptides. Of course, linear block code of this type enables us to calculate with 64 dipeptides. Otherwise, we will have too much elements, square. And we extracted a set which was with significantly different elements. In logistic regression, class standard 1 discriminated perfectly between alphas and betas. The results were reliable, confirmed with SMO. At that time, machine learning started to be available, and we used that SMO algorithm, which is a very good one, and it just confirmed us the situation. It was identical as with logistics. Cross validations were fine, and the classification regression trees were fine. And basically at that time we showed that we could calculate it fast on a PC. It was 2001. It's different today. And another thing is, of course, that we don't need physicochemical parameters to specify protein folds, but we need all information for the nucleotides. 2 years later, we extended this prediction to binary coding patterns. One specific binary coding pattern, and of course we can calculate by coin flipping algorithm the position of each nucleotide on the unit interval. Then we notice the phenomenon that there are two distinct groups of amino acids, and codons, of course, which are characterized, for example, this one was done for Graham polarity scale, but later on we found out that it is almost a rule. It works from Miyazawa-Jerningen contact potential, atom depth, for relative solvent availability. So these two groups cluster. These two groups are incomplete code why. It's you excluded. As I mentioned later, I have to hurry because I'm way off. But we might have, because I have some more time. because the Russians are missing, so okay. By putting y and projections on the unit interval, you can also extract group of codons and a modified group of elements, which also give you information not from those two groups of amino acids, but from the codons itself, of, for example, dipeptides. 8 times 8 is 464. So basically, this what works for amino acids works for codons, works for amino acids as well. With Y, with U excluded amino acids. So you have a cluster with U excluded and the cluster. Interestingly, when you observe standard blossom or other matrices, you also got Y. So it is very conserved within very different measurements and even evolutionary trees. Or matrices for analyzing. Another point is, of course, that with the prediction doesn't start immediately. Within a sliding block, you need a certain number of elements. The rise in the prediction from the frequencies of those elements starts from 3, and it works best with 5 or 6. We have 5 or 6 elements of polarity. You obtain a relatively stable prediction, almost 100% accurate, and cross validation it comes to 90%. What you can do, since it's the triple check code, so you have 3. You don't. The next 3 check bits, you can observe not the check bits, but you can observe the next elements and shift it. Now, shifting the 3 elements of the triple check code, encoding them with the letters, and the next 3 elements, you can observe, for example, 12 amino acid blocks, or even larger. This makes a huge reduction in the number of elements. So you can compress the information for 8 billion times, for example. And even you can reduce in the 12 amino acid window, so you see for 4.10 to 13 to 64 elements. It works well. That was 2003. Now it's going very fast. Nobody cares about old papers anymore. And of course, one of the other characteristics, which I won't go into much details, is that the result, but we will see it on the square of opposition, or by semiotic square, is that due to this representation, we have two types of clustering of coding elements and amino acids with them. One is polar non-polar, which is UA distance. So UA distance, and they differ. They're on the opposite side. While GC distance tend to cluster, make a central cluster, neutral. And that is really the fact. And so-called molecular recognition theory, which claims, and many authors has done that, and the peptide design of interacting peptides uses this evolutionary trick to obtain new tests, interactive peptides, and some biotechnology modeling based on those things. Here, just to tell you is to show you how those opposite polar, non-polar, and neutral, so these are UG complementaries, these are UA, these are GC complementary groups. You can do complements in 3 to 5 or 5 to 3 direction, and of course use it for the tests. This is how it looks. As biosemiotic square, or square of opposition, or probabilistic square. Probabilistic square of opposition, which was done recently, is important because it enables us to calculate the positions within intervals from the empirical data. And here you see Kimura. You just turn Kimura for 90 degrees, And you've got the classic square of opposition. And of course it is the same. You have transition, transversions, and complementarity. Danckwerts and Neubert call that transition, transversion, or is basically complementarity with both procedures. You either flip groups or the rings, or groups and rings. But when you flip groups and rings, you got complementarity. All this was published in Code Biology 1, 2, and 3, and some other papers, but these are basically core papers for this type of concept. Semiotic square holds with the same. And when you go to UCGA, now something resembling Klein-Cross shows, but this is Ulmer. Ulmer emerges. Those black fields are invariant in all, irrespective of the third letter. So, and most of these amino acids are early amino acids, so the early evolution process. When it's UCAG, so when it is coded in gray code, which is not bad per se, but this picture then is not obvious and it's not easy to see. So imagine 4 tables, and these black groups, which is basically a relational model that you will see in a few minutes, goes through all this. The other white parts.
Today we have time to talk about this in minutes. We need another talk.
Okay. Can I have 5 minutes more?
Okay. Okay.
Okay. Thanks. So it's basically a solution to Danckwerts and Neubert problem. When he used Klein groups and they extracted 3 operators and a unit operator. Now we know that according to IUPAC we can give symbolic names, addresses to those operators. One is E, or and transversion, transition, transversion is, and transition are also, I put it, amine and keto groups, so that you could see. So basically we could calculate with codon symbolic and IUPAC positions. Now I'm coming to probabilistic square hexagons. Sanfilippo and Pfeiffer did it. It's a sort of outgrowth of Italian definiti schools. They did a lot of work, and those classifiers can be used from psychology investigations of human behavior to engineering research. They formalized quantifiers on the Aristotelian square of opposition. So it is that probabilistic square of opposition. The important thing is that you can graphically calculate and obtain addresses and the positions of U, C, G, and A. You may even have intervals. So I will skip the continuity principle and all this stuff, and just to say, and the relational model. Now, the relational model makes sense in this context because we wanted to embed modeling and calculations into manipulation of data. Relational databases as such were proposed by Pearson. Pearson did FASTA. So he probably most used bioinformatic language today. But relational databases make more sense when you have a relational model of code. And the relational model fits perfectly with Dankworth-Neubert or Klein four groups model. Why IUPAC? Because, as people noticed, this IUPAC nomenclature also give you ability to analyze some extra incomplete data from the incomplete or partially complete or unspecified amino acid sequences. In the relational model, you have four tables because the third base can be UCGA. So first decode classic table, that's the algorithm. Then make for each third letter its own table. So UNC, but UNC identical. And all groups have a joint, so-called joint table through all four. The joint table is necessary to join all the elements and in the concept. So you have first base, which is row, second base column, third base table. I will skip Klein just to show you analogy. This is schematic representation. So you see B, A, B, and C. A is change in hold ring, change group. B is it's basically transition. Transversion is B, change ring, and change ring and group is C. Probabilistic spherical position, you can have it and calculate. And just to say, the operation resembles XOR, classic, when you divide strong and weak groups, so it's complementarity. In this whole system, with Klein groups, the complementarity is the core part. And we see how it looks corresponding to the amino acid mutation rings. And of course, you can put it into binary. So UCGA, and 1357 is usually for the Klein groups, or you can put 0 and 1s. From this you can calculate two duplex addresses of letter 2 and 1, represented with two symbols and the number, and calculate with those numbers. And you see patterns here. Central core part. This is strong weak base pairs. Okay. And we can, okay, I'm finishing. Calculate from it, this is for purine pyrimidine, and this is for aminoketo. Position it at an interval, calculate it through a square, and obtain a relative surface area or Grantham formula with a fantastic precision from 4 elements, from ring, weak cation amino, 0.94, adjusted S square is 0.84. It's Grantham polarity, empiric surface area, empirically obtained on 4,000 crystallographic data. Atom depth, classic atom depth, and classic statistical potential, which I used. And we see Miyazawa-Jennings and contact potential 0.97. The error is 0.5. Even Miyazawa-Jennings accepts 7%. So it's less than. With adjusted R square is more 0.22. Thank you.
Just Just a minor suggestion about the previous presentation. If you have considered the use of convolutional codes instead of gray codes. Convolutional codes, you have some advantages.
Gray codes are no importance in this matter. It is just a historic discussion on the distribution of letters in the table. Because what really interests us is a proper nomenclature of rings and groups. And accidentally it happens that when the letters were put initially in a table in the 60s, they were put in gray code. So maybe because at that time they were different, they were popular, maybe. And one of the reasons people say that they were doing experiments with nucleobases according to the price. So G was, well, as some people refer to that, G was more expensive than A, so they did A the first. Whether it's accidental or the problems with the letters came later when people in 70s and 80s were trying to put to each nucleotide its amino acid physicochemical values. Then Rosemary Swanson noticed that there is something which has to be decoded. Probably her husband Stan was in a computer and in computing technology, so he also gave her some hints about it. I remember we discussed that in 2004 when Sergey was doing in Florida for one of the first bioinformatics conference of this kind. Anyway, now we have basically a system made on client groups, which is good to produce patterns and which can be calculated. But thank you for the convolutional codes and yes.
Okay. Another question, another comment about anything in this conference? About this last paper, I just wanted to comment that von Neumann architecture, not model, but architecture is considered the real architecture that the general computers that we use today implement. So it is considered an implementation of the Turing machine. In that sense, all the stuff that we have been listening in this conference has a simulation support in the von Neumann architecture. Even if we can discuss, or if we are willing to discuss what is the artificial life, we could simulate it based on the implementation support that the von Neumann architecture already deploys to us. What I did not understand is what are we intending to extend into the von Neumann architecture for supporting all the simulations that biological systems need and deserve. That was my only comment in terms of asking the author to...
Yeah. I think that this got kind of lost because it's not about extending the von Neumann, but extending the mechanism to accept von Neumann and other models like this. And it's still kind of like tinkering about it around this idea, not something exactly like at the point right now. So, yeah, that's it.
But as I understood it, what you're trying to do is like Patty, you want to get the von Neumann model to be able to evolve.
Yeah.
And I think that's very important. And so it has to be done by adding something to it that can change while the core structure information still remains the same. It's the same like in the cell. You have a very conserved coding structure around the ribosome and what Marcello calls the ribotype. Something that Don didn't say yesterday, which I'm going to repeat now. One of the important things that Marcello brought to us is the distinction between not only the genotype and the phenotype, but the interpretant in between, which he called the ribotype. And that's highly conserved, also genetically highly conserved. But there are many other genes. Of course, the whole rest of the phenotype of the cell can change. And that's what happened during evolution. But that core part, let's call it the von Neumann part if you want to, that remains very, very highly conserved. If you change that, then you change the constructor itself, and then you have a problem.
Yeah, I would like to add the difference between mechanisms, because if you go to Rosen defined a complex system as that it cannot be simulated by a model. So this is the definition. In a sense, you have at a certain point a semantic step that gives meaning to what you are doing, that is learning by doing. You cannot do it by doing a representation using a model. So this is the issue. And then why they introduce relational analysis. Relational analysis forces you to define things in different way. And then you have an impredicative relation between this must be equal to this. But then you can no longer make simulation. You can make scenarios. What if I change this? What happened there? How much I can change this in relation to that? They must remain congruent over the functional cycles, but cannot be predicted with a model. This is why I was a little bit uncomfortable with the extended mechanism, because it will not be a mechanism. It will be what is called functional cycle in biosemiotic. So something that managed to keep the meaning of the different coding around. But again, because this is the point, we can do whatever you want with a computer. I can beat the most powerful chess program, but unplugging the computer. That's it. It's that simple. So we have to be careful about getting too much into the coding, because all this is on the notional side. But life has a tangible side, then is regulated by the law of thermodynamics. So whatever we do, we should be aware that there are rules that about what you can do or you cannot do, and what Patti called the epistemic cut, how you go through the epistemic cut, is using semantic. Models do not do that. So this is the old person trying to. Originally I was an engineer, so I'm telling you, it took a lot of years to get into it. But if you're starting now, better to get the immediate.
Well, I'm reacting to the pull the plug issue, and I just I agree that anything that resembles a living organism needs an environment. So we need our planet, otherwise we will not survive, and we may not. But I could imagine that the internet becomes such an environment which is reasonably stable and which will be, and we can destroy it, but we may become so dependent on it that we will not pull the plug. And so,
South Africa.
well, I really think that we shouldn't think that a program lives on a computer. And I also think that, with the internet, models, codes have to become somehow, they have to respect universal standards so that they would immediately do something at any place.
The program will reset the energy. They will mine
Well, you have,
electricity, the programs? I don't think so.
but they will. As long as we
You
maintain.
need some agency at a certain point. Otherwise, the program alone is notional.
Well, it
not
multiplies. It exists in many copies. It exists everywhere in the world. So
Without electricity, it cannot make copies of itself.
no, that's true. But I mean, our planet. Is, no, I'm not sure whether our planet is safe. I mean, we need sunlight, and computers need us to supply.
It's exactly my point. To do code in the genome, you cannot add a pH1. Or you have a rabbit on the bottom of the sea. Is that
Absolutely.
It seems stupid, but very often I don't see this consideration in all these discussions. There will be help a lot. That all these things must have an admissible environment, and because if you are alive, you are not in equilibrium with your environment. Otherwise, you are dead. Okay. So whatever we do is in non-equilibrium thermodynamics. There are rules of that seem to be not present in any discussion that we are having. So this is simply my point, that would help a lot to try to keep in mind this, that all these things happen in biophysical terms in a situation of non-equilibrium, which is not natural in the sense, normally things without life tend to decay. So there is something that is operating to avoid this, and this is something happening in non-equilibrium thermodynamic realm. That there are rules there in Prigogine, Schrödinger. Yeah, this has been the major revolution in science since the 60s, the 70s. Nobody knows.
Nothing to do with code biology.
Yeah, okay. Okay.
Nothing to do with code biology.
I will present tomorrow.
That sort of thing.
My presentation tomorrow is about
Forget me.
looking at the interaction between the two.
Yeah. But let's not go back to the 50s and 60s. No, that's sort of discussion. The long time ago. Yeah.
I will come with the, I will say,
Yeah.
bull. We have fruit
We'll see.
We have a comment here from. Just a small remark, and returning to von Neumann architecture. It has already been extended several times. What we call evolutionary computing or computation has been adopted for some years. Architecture based on what we call field programmable gate arrays, FPGAs, which are devices that have been thoroughly used throughout the world for evolutionary computation. And recently, for the new computers that we are studying, von Neumann architecture is also being extended and rethinking. The new computers that will change completely what we are doing today demand the new approach, and it has already been proved that von Neumann is not the solution for that. So it's not the solution for the new computation, atomic computation and all that. So those that are willing to study theoretical models and implementation models for computation, there is lots of works already published about that. The main extension of von Neumann is called Harvard architecture. Okay, that's all.